Today we will discuss about interactions between mKRAS cell and how they helps in these regulatory cells to either convert them into the M2 type macrophages or helps in this tumor progression. So we will discuss one by one the different or the main ways or main approach this mKRAS used to convert Tregs into the immunosuppression or helps them for tumor progression. So this KRAS release, the KRAS mutated cells release some cytokines, that is IL-33. It interacts with the Treg receptors, Treg cell surface receptors, that is ST2R, and these interactions either helps this Treg to polarize towards this M2 type macrophages or directed them towards this exhausted T cells that does not have any cytotoxicity or any cytotoxic activities, and both of them results in this immunosuppressions. So this is how the IL-33 makes the Tregs to move towards or helps in this immunosuppressions. Other things that it does is it will also release these KRAS mutated cells or the cancer cells generally released some tumor derived exosomes. These tumor derived exosomes then interact with these CD4 plus T cells or CD4 plus CD25 minus T cells, and then it helps this it convert these T cells towards the CD4 plus and FOXP3 Treg like cells, and these FOXP3 upregulations will help in the immunosuppressions. All is known FOXP3 upregulations is known for immunosuppressions. So these CD4 plus FOXP3 cells will help in the immunosuppressions. And the cytokines, that is IL-10 and TGF-beta that are released by this KRAS will convert the CD4 plus T cells towards these regulatory T cells. And these regulatory T cells, by nature, its function is to inhibit the activations of the T cells. So it will help in the immunosuppressions. And these mKRAS mutated cells generally have this IKKα low, and this will result in the release of multiple cytokines such as IL-23, CSF1, CCL22, TNF. And this IL-23 will inhibit the CD8 plus T cells formations. So less cytotoxicity T cells, so less immune activities against the cancer cells. And the CSF1, CCL22, it helps in the macrophage differentiation, and it also helps in the conversion of these CD4 plus, CD25 plus, FOXP3 plus cells towards macrophages, or these macrophages in the presence of ROS toward the CD4 plus, CD25 plus, FOXP3 plus cells. So here CD25 plus cells are there along with this FOXP3. And these cells then interact with this TNF to help in the tumor progression. And or what it can does is it can interact with this TNF to form this FOXP3 regulated T regulatory T cells. It can cause the inductions of this FOXP3 plus T regulatory inductions or FOXP3 plus T regulatory cells, and this will also help in the tumor progressions. So it can help in the tumor progressions with FOXP3 regulations, but enhanced regulation of FOXP3 will also results in this tumor progressions. However, the magnitude of tumor progressions might vary in this case and in this case. And the other things, whenever these mutated KRAS2s are there in the cancer cells, it will cause change in the intercellular signalings. And this intercellular signalings, one of the intercellular signalings that it changes will result in the upregulations of this MAPK pathways. And these upregulations of MAPK pathways will cause release of the cytokines, that is CCL5. And this CCL5 helps in the polarizations towards regulatory T cells or macrophages. That is, it can derive the M1 type macrophages towards M2 type macrophages, and it can derive to the other T cells towards regulatory T cells. And both of these their functions is to suppress the immune response.
So this is how these mKRAS cells and the regulatory T cells interactions occurs, and these are some of the main interactions. Apart from that, some other interactions might also be there that are not presented in this figure. So if you need to understand more about this topic in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.